Warm welcome to the latest OncoShorts podcast, brought to you by MedSynapse. OncoShorts is a weekly program covering a wide range of clinical and scientific medical content that offers enriching insights into the world of oncology. In today's episode, we will be discussing the recent approval of a new drug by the Food and Drug Administration, or FDA, of Zanubrutinib, for adult patients with Waldenström's Macroglobulinemia accompanied with MYD88L265P mutation. The approval came after the results from the Phase 3 ASPEN trial were announced, comparing Zanubrutinib to Ibrutinib for the treatment of Waldenström's Macroglobulinemia. The MYD88 is an adapter protein that coordinates multi-subunit signaling of most of the toll-like receptors and receptors for interleukin-1 and interleukin-18 cytokines. Recurrent MYD88L265P mutation is a somatic mutation and was first found in diffuse large B-cell lymphoma, and further studies have indicated that the frequency of MYD88L265P mutation in patients with Waldenström's macroglobulinemia is about 87%. Zanubrutinib has been investigated for the treatment of patients with MYD88L265P mutation. It is a small molecule inhibitor of Burton's tyrosine kinase, or BTK. BTK is a key component of the B-cell receptor signaling pathway and is an important regulator of cell proliferation and cell survival in various B-cell malignancies. Zanubrutinib blocks B-cell receptor-induced BTK activation and downstream signaling, leading to growth inhibition and cell death. It is an orally active inhibitor that covalently binds to BTK, resulting in the inactivation of the enzyme. The ASPEN trial was a randomized, Phase 3 trial in 229 patients with Waldenström's macroglobulinemia, conducted in 61 centers in Europe, Australia, and the United States. In the study, patients in Cohort 1, including 201 patients, were randomized to receive Zanubrutinib 160 mg twice daily or Ibrutinib 420 mg once daily, until disease progression or unacceptable toxicity. Cohort 2 included 28 patients with MYD88 wild-type mutation and received Zanubrutinib 160 mg twice daily. The major efficacy outcome that was considered for the approval was a very good partial response in the patients, which was assessed by an independent review. The results from the trial were announced after a follow-up of 19.4 months. Very good partial response in the overall patient population was 28.4% in the Zanubrutinib arm and 19.2% in the Ibrutinib arm. None of the patients achieved a complete response in either arm. 12-month progression-free survival was 89.7% in the overall population of Zanubrutinib and 87.2% in the Ibrutinib arm. Overall survival was 97% in the Zanubrutinib arm and 93.9% in the Ibrutinib arm. Adverse events of greater than grade 3 were 58.4% in the Zanubrutinib arm, while it was 63.3% in the Ibrutinib arm. Adverse events identified in both groups were atrial fibrillation, minor bleeding, major hemorrhage, and diarrhea. Neutropenia was observed to be higher in Zanubrutinib arm, accounting for 29.7%, compared to Ibrutinib, which reported 13.3%. Although the trial did not achieve statistically significant complete response, very good partial response rate was higher for Zanubrutinib compared to Ibrutinib. Improved safety and tolerability were also observed for Zanubrutinib comparatively. These factors were significant in the FDA granting approval to Zanubrutinib for patients with MYD88L265P mutation. During the announcement of the results, Konstantin Estam, M.D., principal investigator for the ASPEN trial, stated that Waldenström's macroglobulinemia is a devastating and incurable disease with significant morbidity. The meaningful results from the ASPEN trial help advance the understanding of the role of BTK specificity and off-target effects during treatment. This concludes our today's OncoShorts podcast, brought to you by MedSynapse. Thank you for listening.